All right. We're up on the top of this mountain now that we have seen how proteins are produced, how they take on a shape, and how that shape makes it possible for you to do things with your proteins. And we've talked about viruses and talked about the immune system. And the last thing that we covered was mutation. These first couple of slides are kind of a review from our previous meeting. Recall that you should know how DNA replicates, right? Since DNA is two strands of nucleotides that pair up with each other by the rules of base pair complementarity, right? Chargaff's rules. If you know the order of bases on one strand, you know what the order has to be on the other strand. And so when you double your DNA, the DNA double helix unzips, the two strands separate, and each one can be used as a pattern to build a complementary strand. Okay, that should not be new to you. What's actually doing the work is a set of enzymes called DNA polymerases. That's what causes the DNA to unzip and welds new nucleotides together to make complementary strands. DNA damage is repaired by a different set of enzymes, and both of these are good, but they're not absolutely perfect. Sometimes they make mistakes. Sometimes it's just as simple as changing one nucleotide. You know, you might have one piece of your DNA that goes GAACC, and after one cell divides, it becomes GATCC or something like that. Sometimes this has no effect at all. Sometimes it has a pretty noticeable effect on the shape and the function of the protein that is produced. There are cases where a gene may even be silenced. One single change in nucleotide might be enough to stop that gene from getting transcribed and translated. This particularly happens if a mutation results in a stop codon being placed at the wrong point. You know, if you put the stop codon in the middle of the protein, it will, see what I mean? It'll cut off in the middle and it may not do anything. You can also have, because of crossing over errors in meiosis, you can have portions of genes or even entire genes that can get deleted or that can be duplicated. And all of these are what we consider mutations, changes in the order of DNA bases. And anything that damages DNA, we call a mutagen, because it's capable of causing mutations. And there's lots of things that will damage DNA. There are chemicals that will do it. Radiation can do it. X-rays can do it. Ultraviolet light can do it. All of these damage your DNA. You have got repair enzymes that can fix the damage, and they're good, but they ain't perfect. Sometimes the damage is not repaired perfectly. What you're looking at here is a dish containing a jello-like substance called agar that's got nutrients in it. And each one of those spots is a colony of bacteria growing on the dish. Dishes like this are used in what we call the Ames test. If you have developed some new chemical and you want to put it in people's shampoo or something like that, the first test that you would do would be to expose bacteria to your chemical, grow them on plates like this, and look for particular types of mutation. Because again, DNA is universal. Anything that disrupts bacterial DNA has a decent chance of disrupting yours. It's not a 100% accurate test. You would still need further testing, but it can be done very cheaply, very quickly, It can be done without, harming mice or rabbits or anything like that. And so this is the first line test for mutagenic activity in new drugs, new consumer products, your lip gloss and eyeshadow, if you wear any, or your deodorant, if you wear any of that. Hope everybody does. Was probably tested at some point using the Ames test, or at least chemicals in it were tested that way. A major reason why this matters. We've talked about proteins that carry out chemical reactions. Those are the enzymes. We've talked about proteins involved in structure, like the proteins that make up your hair or the proteins that make your muscles. There are whole families of proteins that are involved in regulating what the cell is doing. This is a much bigger topic than I can possibly cover. You're just getting a little taste of it. This is the Cliff Notes of the Cliff Notes version. But there's a set of specialized proteins that you can think of as accelerators and brakes of cell division that ensure that cell division happens when it's supposed to, that ensures that cells that you don't need more of aren't dividing, and cells that you do need more of are actively dividing. So again, best thing is just to think of these as brakes and accelerators. We don't have time for more detail than that. There's a second process that's also regulated by a set of genes. When cells are damaged, or in some cases when they're no longer needed, they undergo a very orderly process of turning on the lights, mopping up the bar, and putting the bar stools on the tables, shutting down, and ultimately breaking up into tiny little blobs. It's called apoptosis. It's also sometimes called programmed cell death. Here's an example. When you were a little embryo inside your mama, roughly the size of a bean, your tiny arms and legs look like paddles. Now, of course, you have separate fingers. And the reason why you have fingers and not paddles is that the cells that used to be in between your fingers underwent apoptosis. They all collectively got the message to shut down and break up into little pieces, and the tissue in between that used to be in between your fingers just faded away. The cells that made it up broke apart, and now you have separate fingers. And if that didn't happen, you'd have webbed fingers, of course. This also happens in things like if you've ever watched tadpoles develop into frogs. The tadpole tail ultimately breaks down and disappears because of apoptosis. The cells in that tail get the message to quietly commit suicide, and they do. And that's a very important process as well. How do they do this? Again, you could teach sizable upper division courses in this, and some people do. The very general picture is that some genes code for proteins, and the proteins can come back to the DNA and bind to the DNA and either encourage or block other genes getting transcribed. We call those genes either promoters or repressors. And this is a model of what it would look like. The green thing is a DNA binding protein. You can see it's got a lot of alpha helixes in it. And it's shaped in such a way that it can bind to the DNA helix, which is shown here in blue and red. And this particular one can block another gene from getting transcribed. So the upshot is that, a cell can create one protein through transcription and translation, that protein can come back to the DNA and turn on another gene, encourage it to get transcribed and translated, or stop another gene from getting transcribed and translated. So genes can act as on-off switches for other genes. That is as much as I can tell you of the grand mystery of how genes can control cellular processes. Genes can switch each other on and switch each other off. We don't have time to go into this, but because of this ability, that some genes act as on-off switches for other genes, we have these networks of gene circuitry that are diagrammed in this diagram right here, which has a whole bunch of abbreviations for different genes that you certainly don't have to know. Suffice it to say that some genes will promote, act as an on switch, or repress other genes. And those genes that are promoted can act as on-off switches for yet other genes. And then those genes can turn on or turn off yet other genes. And that creates these networks of circuitry. You know, imagine some kind of complicated circuit board where if you flick one switch, that switch turns other switches on or off. And then that set of switches turns yet other switches on or off. So genes are able to regulate other genes. And cells, by the way, can also do this sort of thing in response to signals from other cells. So one cell can send a chemical message to another cell that basically says, switch on one gene. And then when that gene is switched on, it might switch off 2 genes and switch on 3 more. And then the 3 that are switched on might switch off 7 more and switch on 8 more and da. Okay, genes can turn each other on and off. What you're looking at, by the way, is this complicated diagram. Each one of these abbreviations, like OTX and BLIMP1 and WNT8, those are all abbreviations for different genes. And these are the genes activating and deactivating each other during part of the development of a sea urchin embryo from fertilized egg to free swimming larva. This is the kind of circuitry that goes on in the development of an embryo. That's not a sea urchin, that's a mouse embryo that's been stained with several fluorescent stains to pick up different cell types. You can see the nervous system has been stained green. So you can see the spinal cord and nerves branching off the spinal cord at this stage. At this stage, when the mouse embryo is roughly the size of a pea, other types of cells have been stained in blue and red and white. Ultimately, this ability of some genes to switch other genes on or off is the ultimate secret for how a single fertilized egg cell gives rise to a complex adult body with lots of different cell types. The study of this process is called developmental biology, and we just don't have time to get into it further, which is a pity. It's absolutely lovely. What I'll say is that when you develop from a fertilized egg to a fine upstanding adult, that egg contains all of the DNA that all of your cells will ever need. As your cells divide, they do what we call differentiate, which means they become different cell types. Your liver cells and your skin cells and your muscle cells and all of the others ultimately come from that fertilized egg. And what happens as that fertilized egg cell divides and divides and divides again is that cell types develop by shutting down the genes that they're not going to use. your muscle cells still contain the DNA that they could use to produce liver proteins. The reason why they don't is because those genes they're still there, but they've been turned off. They've been repressed. They've been deactivated. And the cells in your liver have all of the DNA that's needed to make muscle proteins. And the fact that they don't means that those genes have been deactivated. And so there's this pattern of gene regulation that ultimately results in you having different types of cells to do different things. And it's all got to do with these gene regulatory networks that are absolutely beautiful and awesome, and we just ain't got the time. Another reason why this matters is, remember I mentioned that we have these genes that can act as brakes or accelerators on cell division. Any mutation that hits them can cause the cell to start dividing uncontrollably, like a car with a stuck accelerator and cut brake lines. Just like a car with an accelerator that's stuck and no brakes is going to keep going until it eventually hits something. The same is true for a cell that has damaged oncogenes. The cell may start dividing uncontrollably. Now, apoptosis is a kill switch. If you have a cell with damaged oncogenes that starts dividing uncontrollably, the cell can basically commit suicide. It'll blow itself up. But if the switch that regulates apoptosis is also mutated, the cell won't be able to self-destruct. It will keep going and keep going and keep going. And the end result is cancer. Cancer is not really one disease. There are maybe 100 different types of cancer. That's partly why there's no single cure for cancer, is that there's no single type of cancer to be cured. There's lots of different types of cancer. But the fundamental thing that goes wrong in all types of cancer is cells are dividing and they can't control the division, and they can't stop dividing, and they can't self-destruct. You end up with, if your dividing cells make a mass, a big clump in the body, we call that a tumor. Other types of cancer, like leukemia, there aren't any tumors. The cancerous cells, as they grow, they can press on important structures like nerves or blood vessels, especially if they're in the brain, where the brain is locked inside your skull. And if something starts growing in there, the brain gets damaged by the pressure because there's nowhere for the brain to go. Cancers can also divert the body's energy to themselves. Cancers can make blood vessels grow into the tumor and feed it with, your body's nutrients that the rest of your body needs. And cancers, of course, can bud off and produce new tumors at other parts of the body. And that's ultimately why it's bad for you. And anything that damages DNA, which is which we call a mutagen, is a potential cause of cancer, or as we say, a carcinogen. Okay, somebody's getting kicked out because of unstable internet. My sympathies. I can't fix this from here, but good luck with that. I hope that clears up. So anything that damages DNA is a potential cause of cancer. And anything that damages DNA, by the way, for the same reason, is a potential cause of birth defects, is an embryo toxin. So X-rays, for example, can not only damage cells enough to cause cancer if you're overexposed to them, they can also ultimately cause birth defects if they mess with that, gene regulatory network that's going on in embryonic cells. That's why we need to know whether something causes a mutation or not. And incidentally, you can inherit faulty oncogenes, and this is why some types of cancer tend to run in families. You don't really inherit cancer, but you can inherit oncogenes that are more prone to mutation, that may not work as well as they should. One example of this is BRCA1. If you inherit that gene, you're at an increased risk of developing breast cancer. So that's why some types of cancer can run in families. Some types of cancer are more likely if you're exposed to mutagens like radiation or certain chemicals or things like that. And sometimes cancer is just a result of bad luck. Everybody has mutations. DNA replication is good, but it's never perfect. And some people do everything right and still end up with cancer just because DNA replication is not perfect. And every time a cell divides, the chance of a mutation hitting the oncogenes is very small, but it isn't zero. So some types of cancer are inherited. Your risk of cancer goes up if you're exposed to mutagens, and sometimes it just happens because it happens. Right. Not all mutations, be it said, will lead to cancer. Many of them change the primary structure of a protein. Remember, that's the order that the amino acids are lined up in. And potentially they could change the shape of the protein. Some mutations have no effect at all. You might recall when we had that chart of the genetic code that most amino acids are coded for by more than one codon. So I think UUU and UUA both code for phenylalanine, if on the messenger RNA. Meaning that fairly often a mutation that just changes one nucleotide will change the nucleotide, but it won't change the order of amino acids. Because you go from a codon that stands for one amino acid to a different codon, but it stands for the same amino acid. And by the way, we call that genetic code redundancy. So mutations that do that simply don't do anything. They have no visible effect. Even mutations that do alter the order of amino acids, sometimes that doesn't matter. Sometimes that doesn't change the protein shape in any way that's important. If we think of proteins as being like tools, some mutations would do things like, change the color of your screwdriver handle from blue to pink. Okay, that doesn't have any effect on what the screwdriver does. The color doesn't matter. And the same thing is true for some mutations. They might alter the shape of a protein, but not in any way that makes a difference. And probably the great majority of mutations are what we call neutral, meaning that they're neither bad nor good. They just kind of happen. We're all carrying mutations, by the way. Every human being has, sorry, brief mental calculation, something on the order of 10 to 20 spots in your DNA that are altered from what your parents gave to you. We're all mutants in some way. Most of them don't do anything, or if they do anything, it isn't important. Are there ever mutations that are beneficial? Well, you usually can't answer the question without looking at the environment, but there are cases of beneficial mutations. Although whether it's beneficial or not depends on what the organism's environment is. Let me show you an example. That's Dr. Richard Lenski at Michigan State. And that's one of these dishes I was talking about with a jelly-like substance inside called agar that you can use to grow bacteria. And Dr. Lenski has been growing a bacterium called Escherichia coli since 1988. And the nice thing about E. coli is that if you give it normal, decent conditions, it can divide 6 times a day. So as of a few years ago, they had been able to observe E. coli changing over 76,000 generations, right? If you go through 6 generations a day, then since 1988, that's what, 30 years, works out to about 76,000 generations. That's a lot. So they can look at change over, long spans. What he's found, if one of you could mute your mic, that'd be great. What Lenski found is that roughly 1 in 12 of the mutations turned out to be beneficial for the bacteria. One particular set of mutations that he was able to document gave the bacteria the ability to use a food source that they previously couldn't. The bacteria had been growing on a sugar called glucose, and he switched them over to a different food source, a molecule called citrate. And at first, the bacteria couldn't use it much, but as the bacteria grew, they underwent mutations. The mutations created different varieties of bacteria, and some of those varieties of bacteria developed the ability to use this new food source. So he's actually been able to see mutations happening that turned out to be beneficial for the bacteria because they gave the bacteria this ability to use a different molecule for food, which previously they couldn't. Does this ever happen in nature? 1935, chemists invent nylon. I'm assuming you've heard of nylon, used for everything from parachutes to pantyhose, and produces some waste chemicals. And there was a factory in Japan making nylon that had a pond out back where they kept waste water, which contained a lot of the chemical byproducts from the manufacture of nylon. 1975, Japanese scientists discovered a bacterium living in that wastewater pond. It hasn't been given a proper species name. It's called Pinarthrobacter K172. And what's cool about these bacteria is they can digest the byproducts of nylon. They can eat nylon waste products. And here's the kicker: nylon didn't exist before 1935. Before nylon was invented, those particular chemicals didn't exist on Earth, as far as we know. Somewhere between 1935 and 1975, a strain of bacteria just happened to undergo mutations that altered their enzymes in such a way that they were able to break down nylon waste products, which they were not able to do before. So hint, hint, we're back in natural selection territory, and we've finally got an answer to Darwin's dilemma of where does variation come from? Ultimately, it comes from the fact that living things duplicate their DNA very well, but they don't do it perfectly. Mutation is the ultimate source of new alleles. And some mutations give you alleles that don't promote your survival and reproduction, and those get lost. And other mutations give you alleles that do turn out to be useful. And the easiest organisms to observe this have been in bacteria. Lenski's long-term experiment has been giving us quite a lot of really cool data on this matter. But we can see cases of it happening in nature as well. Does it ever happen in humans? Do we ever get human mutations that are beneficial? Well, we don't exactly get the X-Men. That's a comic book. That's made up. And anyway, I seem to recall Wolverine's not technically a mutant. He's just had adamantium bonded with his skeleton. Maybe you can correct me on that. So we don't quite get this sort of mutation. But here's an example that those of you that play sports might like. This gentleman was an Olympic athlete at the Winter Olympics in Innsbruck, Austria, way back in 1964. His name is Eero Montiranta, representing the country of Finland. He was a cross-country skier. That's one of the most physically demanding sports that there is. And he won 2 gold medals and a silver, just left everybody in the dust. Didn't even particularly train that hard. He was just insanely good at winning cross-country skiing races. Turned out he had a mutation in a gene called EPOR, erythropoietin receptor. That's a protein that's involved in regulating the number of blood cells that you make. It regulates the process of making blood cells. If you must know, making red blood cells is called erythropoiesis. And this protein that's involved in regulating it is erythropoietin receptor. And so did 5 generations of his family. I don't think you've done this in lab, but I do believe you're going to in the last lab. This is a family pedigree chart. And he's right there. He's represented by that square number 17. In family history charts, males are represented as squares and females are represented as circles. So he married his wife and they had a daughter and then a pair of twins, a boy and a girl. And then that square right there represents his father. That's his sister. That's another sister of his, and so on. So they took Montiranta's family history and traced it back 5 generations. This arrow right there just leads to more of the family history chart because it was too big to put all on one slide. But that's his family. The slash means somebody has died. And the plus or the minus right there represents whether the person has this EPOR mutation. People with this mutation make extra red blood cells, and that means their blood is able to carry more oxygen. You might have heard of blood doping, where athletes will extract some blood, take out the red blood cells, and then put those red blood cells back into their blood just before competing. That's illegal. But Montiranta was basically naturally blood doped because his body just made extra red blood cells. There are people that do this naturally and they can suffer some health problems, like an increased risk of stroke. But for whatever reason, that never happened in Montiranta's family. Everybody was perfectly normal, except the people that had this mutation were able to pack on extra red blood cells. And that's his family right there. That's him sitting down there next to his wife. And then you can see his, I don't know who's who, but you can see his kids and his cousins and his nieces and nephews and all of that. And yeah, nobody in the family suffered bad effects. Nobody died young or anything like that. The only difference was their blood could transport 50% more oxygen than a typical person's blood, with the result that people that inherited this gene had incredible endurance. So that's a case of a mutation. It happened at least 5 generations before Montiranta. How far back it happened, we're not sure. And what it did was, in fact, convey, maybe not as cool as Cyclops' optic blasts, but it did, in fact, convey a superpower. Montiranta was naturally very, very good at endurance athletic events. By the way, in case it wasn't clear from the last lecture, the reasons why we keep getting new strains of virus is mutations. Virus DNA is subject to copying errors because any DNA is. Most of those errors don't have an effect, but a few, just by dumb, rotten, stupid luck, may enhance the virus's ability to spread or its ability to dodge the human immune system. And we've seen this since May 2020. You know, we had the initial coronavirus infection. That strain got replaced by a new strain called the Alpha strain, and more recently by the Delta variant, and possibly by various others that pop up every so often in the news. That's just because every time that a virus replicates, there's a chance that that virus is going to pick up a mutation, a change in the order of its DNA, its nucleotides. And it's a small chance, but it ain't zero. And if you get infected with COVID, you end up with, you know, hundreds of millions of virus particles in your body. And the chances are very good that at least some of them are carrying a mutation. Most of those mutations might not do the virus any good. Most of those mutations either do nothing or might actually weaken the virus. But every now and then you get one that makes the virus just a bit stronger, just a bit better able to infect, which is not good from the point of view of humanity, but it's certainly very beneficial from the point of view of the virus. The same thing happens with the flu, and this is why you keep having to get a flu shot every year, because the flu that's going through this winter is not the same as the flu that went through last winter, because flu is constantly throwing up new mutations. It's also part of the reason why it's been so hard to develop an HIV vaccine. HIV mutates so frequently that the human immune system can't adapt, and any vaccine that you got would not be guaranteed to be effective against later varieties of HIV. HIV changes faster than either our vaccine developers or our own bodies can adapt to change with it. And I think I showed you these slides before, but just as a reminder, the Alpha variant of COVID-19 has 3 points in its DNA where there's been a deletion and 13 other changes in its nucleotide sequences compared to the first strain that we found. That makes it more infectious. It turns out that so far it hasn't made it able to bypass the protection of the vaccines, but there is worry that at some point we are going to see a variant of the COVID virus that can dodge the protection that vaccines can give. One of the mutations in the COVID spike protein changes amino acid number 501 from the amino acid asparagine to the amino acid tyrosine. That one little change right there, shown by the arrow, seems to improve the virus's ability to bind to cell surface proteins. The virus sticks to your cells better and is more effective at entering into the cell. So sometimes all it takes is one single change of one amino acid to another amino acid to cause a very noticeable change in how the protein functions. And in this case, it improves the protein's ability to bind to cell surface proteins. That's not an improvement from our point of view, because we don't want viruses to get better, but they do. And again, the Delta variant has yet more variations in those spike proteins that increase its infectivity. Now, you might be wondering how it is we can read virus DNA when it's way too small to see. What you're looking at there is a little snippet of coronavirus DNA. There is a part of coronavirus DNA that starts with ATG and then TTTG, TTT, TT, CTTG, TTTT, ATTG, CC, ACTA, GT, CT, CT, AGTC. I hope you're memorizing that. I'm kidding. Don't. And you might be wondering how we know, what the order of bases can possibly be on any given piece of DNA. Well, to work that out, we have to look at the other great scientist that you know, who got his PhD from the University of California at Berkeley. I'm the one that you already know about, but let me introduce you to Dr. Kary Mullis, biochemist, surfer dude, and connoisseur of recreational neurochemicals. Okay, the guy took acid and shrooms a lot. Let me put it that way. And he was working for a biotech company in the San Francisco Bay Area, great place for surfing too, back in the 1980s. He got the idea in 1985, according to his autobiography called Dancing Naked in the Mind Field. And he got the idea, inspired in part by dropping acid, for a technique where you could take any piece of DNA and make millions of copies of it. To be fair, there were other people that had come up with similar ideas before, and these days, we may have this mental image of a scientist as being, a lone wolf up late in the lab, working on something entirely on his own, like Victor Frankenstein or in his laboratory or something like that. The truth is that these days, just about everybody works as part of a team, and Mullis certainly did as well. But he was the prime mover in getting his idea to work, and he ended up winning the Nobel Prize in Chemistry in 1993. He did go on to write this book, and he's kind of gone on to espouse a number of kind of weird causes that probably aren't very good science, and I assume were probably inspired by, talking to the mushroom people or something like that. He's certainly an interesting character, and I'll just leave it there. But he did have one good idea, and the technique that he developed is called PCR, which stands for the polymerase chain reaction. And yes, it's done in tiny little plastic test tubes with caps, like those. And it goes like this. This is a bacterium called Thermus aquaticus, and it happens to be an extremophile, which is a word that we used way back at the beginning of the class in maybe week 2 or 3, when I was talking about archaeans and things like that, way back when. And an extremophile is an organism that is adapted to live in an environment that most living things can't survive in, that's too cold or too hot or too salty or too acid or too alkaline. For most other life to survive in. Well, one of these extremophiles is a bacterium called Thermus aquaticus. It looks like this, and it happens to live at high temperatures. Here's the thing. The reason why you can't live for very long at very high temperatures is because, like, if I was to stick you in water that was nearly boiling, I'm sorry, don't think about that too hard. But aside from the pain and things like that, one of the things that would immediately happen would be that all of your proteins would start getting denatured by the heat. The heat would force them to change shape. All of your proteins are adapted to work within a particular temperature range. And if I make any part of you too hot, those proteins lose their shape and they start changing shape, and that means they lose the ability to function. Your saliva breaks down starch because it's got a protein in it called alpha amylase. But if I were to boil your saliva and then put it on some starch, it wouldn't do anything. Your saliva is just not adapted to work at that high temperature. But the proteins of Thermus aquaticus can work at that high temperature. They're adapted to operate at high temperatures. So you take DNA and you heat it, and heating DNA causes that DNA to unzip, and it breaks those hydrogen bonds that hold the strands together. You go from a double-stranded molecule to 2 single strands. You can unzip it with heat. Then you add the DNA building enzyme that you've extracted from Thermus aquaticus. You add that DNA polymerase that you got from the bacteria, and that enzyme is, which is shown here in a model, is perfectly well adapted to do its work at this high temperature. So the Thermus aquaticus DNA polymerase builds new strands, and then you cool the DNA, and then you heat it again, and the DNA separates, and the Thermus aquaticus polymerase builds new strands, and then you cool the DNA, and then you heat it again. You basically heat and cool your DNA on a carefully timed cycler. And if you repeat the process, you'll end up with millions of pieces of DNA that should all be identical, or nearly so, to the DNA sample that you started with. There's a little more to it than that. That gives you the basic idea. And you can modify the procedure a little bit. We don't have time to get into exactly how this works, but that's what Wikipedia is for. Means that you can modify PCR and make it possible to sequence DNA directly. You can take one little piece of DNA, copy it millions of times over, separate the fragments out using a process called gel electrophoresis, which you're going to see in the last lab that we do, and read the order of nucleotides. This picture right here probably just looks like a mess, but you read it from left to right, and it's almost like reading music. So what this is saying is that the order of nucleotides in a piece of DNA is G, G, C, T, T, C, G, A, G, 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 A, C, T, A, C, A, and so on. And again, it's almost like each one of these little black squares right there is like a note. In fact, you could sing this. You know, you could represent this DNA as a sequence of musical notes. You could go, you know, G, G, C, T, T, C, G, A, G, 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 A, C, A, so A, C, T, A, C, A, G, 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 A, G, C. So on. That's my attempt to sing DNA. And there are composers that have actually experimented with that, you know, putting pitches to DNA bases and turning DNA into songs. I don't know, any music majors might want to try that sometime. Why not? And that's what made it possible for, oh shoot, what was her name? For forensic scientists to identify DNA samples from individual people, even if you've only got a tiny amount of DNA, you know, one little swab from a crime scene with somebody's blood or saliva or other fluids or what have you. Yeah, Abby. Okay. Abby from NCIS. Okay, I was trying to remember the actress's name. Okay, Abby Shuto. I can't remember who played her, but yeah, I always thought she was really cool. And this is what makes it possible for Abby to do all of that stuff. Okay, Paulie Perez, thank you. Okay, this is what made it possible for Abby to do all that stuff that secretly impressed her boss, even if he didn't really express it very much. We can use a set of techniques called DNA fingerprinting, and there's several ways to do this, and I can't cover all of them. No, but the most common method that we use today for identifying a DNA sample takes advantage of the fact that not all of your DNA codes for proteins. Large parts of your DNA are involved in other ways in controlling, regulating your genes, but they don't directly code for proteins. And there are some parts that don't do anything. And one set of these parts that doesn't do anything are called STRs, short tandem repeats. And an STR is just a section of your chromosome where the nucleotides repeat a short sequence over and over and over. So there might be some part of one of my chromosomes that just consists of a repeated CATG, 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 CATG. And those vary in length. There might be an area on one of my copies of chromosome 1 where I've got CATG repeated 8 times. And one of you on your chromosome 1 at the same spot might have CATG repeated 12 times. And another one of you, it might be 6 times. And another one of you, it might be 15 times. And another one of you, it might be 3. These STRs vary quite a lot in their length. And they don't particularly, most of them, certainly the ones that are used in forensics, don't directly affect human health because they don't do anything. But what we can do is take a DNA sample, make lots of copies of it with PCR, and use a technique called gel electrophoresis. You'll be doing this in, I think, the last lab of the year. So I think in like 2 and then 2 weeks, in a couple of weeks, you should be doing this yourselves. What gel electrophoresis is basically just a way of measuring the size of a piece of DNA, measuring how long it is. And we can use gel electrophoresis to compare the STR lengths in an unknown sample with the STR lengths in a known sample. So to do this, and you'll be doing this yourself soon, you take samples of DNA fragments and you put them into little wells. These are just little rectangular holes in a gel. And the gel is very soft and flexible and tears easily. It's roughly the consistency of Jell-O, but unlike Jell-O, it's made of a type of carbohydrate called agarose. You put DNA samples into a gel, and then you plug the gel into a rig that applies an electrical current. You put electricity through that gel. And DNA has a slight electrical charge, and it's attracted to one end of the gel by the electrical field. So DNA gets physically pulled through the gel. Small fragments of DNA pass through more quickly than large fragments. So what you end up doing is sorting all of your DNA fragments by their size. So we can use this to measure the size of pieces of DNA. Even if we cannot see the DNA itself, we can take like a swab from a crime scene, copy the DNA in it many, many times over. That's called DNA amplification, and then sort the size of the fragments that we get by using this gel electrophoresis. So here's the thing. So we can use this technique, and it works fairly simply to sort DNA by size. That means we could take the DNA from my chromosome number 1, let's say, and determine, how many copies of that CAGT sequence it had. For any given one of these STRs, these short tandem repeat ranges, 5 to 20% of humanity will have the same length. Let's say I've got 8 CAGT repeats on my chromosome number 1. 5 to 20% of people will also have 8 repeats. But there's lots of STRs on human chromosomes, many of them. And if we try to match people based not on the length of just one STR, but on the length of 6, 8, 10, 12, it's much less likely that 2 people will just happen to have exactly the same lengths of all these STRs. The SDI's DNA database called CODIS, when they take a sample of your DNA, the information that they store is the length of 13 core STRs. More recently, they've started storing data on 7 more for 20. And the probability of 2 people who are not related to each other just happening by chance to have all 13 STRs match up is 1 in a quintillion. The quintillion is a 1 with 15 zeros after it. It's a very big number. So it is not completely impossible, but 1 quintillion is something on the order of 100,000 times larger than the entire population of humanity. It is vanishingly impossible that somebody who is not related to you could possibly have exactly the same lengths of all of those STRs. This is where those STRs are located. Don't worry about memorizing these names. Let's just say that what each one of these black and white stripy things is a diagram of a different chromosome. And on chromosome 2, there happens to be an area called TPOX. And that area of TPOX happens to have some short sequence that varies in the number of repeats that it's got. I might have, 12 repeats at the position called TPOX, and you might have 9. And then on chromosome 3, there's an area that's just called D3S1358. And I might have 9 repeats and you might have 6. And then on chromosome 4, there's a site called FGA, and maybe I've got 15 repeats and you've got 9, and so on. These are where these 13 STRs are located. And when we measure their length, this is the kind of fingerprint that we get. This is a person who's got, for example, he's got 2 copies of chromosome 19, one of which he got from mom and one of which he got from dad. One of his copies of chromosome 19 has an STR called D19S433. One of his chromosomes has 13 repeats at that location, and the other one of his chromosomes has 14 repeats. Okay, over here on chromosome 12, he's got an area of chromosome 12 that's called VWA. And at the VWA location on chromosome 12, one of his chromosomes has 14 repeats, and the other one has 17 repeats. And then over here, he's got TPOX, and it just so happens that he has, both of his chromosome number 2s happen to have 11 repeats at the TPOX location, and so on. The small hills just before the spikes, probably what is going on is that most of the guy's cells, like here, might have 17 repeats, but he might have a mutation that gives a few of his cells 16. Or he might have a mutation that gives a few of his cells 12 instead of 13 repeats or something like that. So yeah, you do occasionally get interesting little things like that going on. But this is basically how you make a DNA fingerprint. You're essentially counting the number of repeats of a short, meaningless sequence in somebody's DNA. And why, yes, thank you, that is really cool. The technology to do this started becoming available in the 90s. Once the polymerase chain reaction was not only worked out, but equipment is made so that it has become nearly automatic. You can almost, take a sample, put it in one end of a machine, and spit, out come the results in a couple of hours. And there have been worries about privacy issues, and there's definitely ways that DNA evidence can be misused. It's a tool, and there are certainly ways that you can misuse any tool. But I want to end on a slightly higher note, because DNA evidence, one of the things that you can do with it is overturn wrongful convictions. And as of April of this year, 375 prisoners, including 21 who were going to be executed, have been exonerated by DNA testing, by lawyers taking their case, going back to DNA evidence that had been stored from the original crime scene, getting that DNA tested, because either it, often because that evidence was collected before DNA testing could be done, or alternately it was collected but never tested because the defense attorney was an idiot, or the prosecuting attorney knew people who knew people and played golf with the judge and kept it from being tested. Anyway, they were able to get that DNA evidence retested, and were able to demonstrate that the person that had been locked up could not have committed the crime. So these two gentlemen, Mr. Brooks and Mr. Brewer, were freed in 2008 after having been in prison for years for rapes and murders of children that they did not actually commit. So there are ways that you could abuse this, but there are certainly ways that you can use this technology to, you can't give people like this their lives back, but you can at least give them some measure of justice, some measure of satisfaction, and sometimes find the actual criminals, not people who were wrongfully convicted the first time. So it's a tool. There are ways you could use it for evil purposes, and there are ways that you could use it for good. And this happens to be, as far as I'm concerned, a very good purpose. And on that note, I will go ahead and and stop the recording right there.